But with that, I would like to introduce our next speaker. We're diving into our native seed production presentations, which is the theme of today. And today we are beginning with Pat Reynolds and Haleigh Holgate with Heritage Growers. Pat and Haleigh, you can turn on your video and share your screen when you're ready. And they'll be speaking with us today about collecting and amplifying source-identified seed for CALTRANS Willets Bypass Wetlands Migration Project. Or maybe that's mitigation. But Pat, if you're back from the break, you can now share your video and start your presentation.
Okay, very good. Thank you so much. Thanks, everyone. That was so inspiring. Just want to thank Secretary Howland and Director Stowe Manning, and particularly Jennifer Jewell for that such inspiring presentation today. So, okay, very good. So today we're going to talk to you about a really interesting project. It's the CalTrans Willets Bypass Wetland Mitigation Project and all of the seed work that's been done on that with Heritage Growers here. But before I do, I just want to take a minute here and tell you a little bit about Heritage Growers and what it is that we're trying to do to solve the source identified native seed bottleneck. So at Heritage Growers, we are a native seed and plant supply company providing source identified native seed to support habitat restoration in California. We're a nonprofit organization that's part of River Partners, so that makes us very unique. As a result of that, any profits that we generate at Heritage Growers goes back into the mission of River Partners of restoring habitat for the benefit of people in the environment. We have about 168 acres currently in production, including about 130 different items, mostly native grasses and native forbs, all of which are source identified and restoration appropriate. We have a demonstration garden that we use for lots of different important uses. We use that to provide seed for us for our nursery program. We do small plot amplifications there. We collect data on plant phenology, which then informs both our farming practices as well as the advice that we give to our partners and collaborators when using our seed for habitat restoration. We also have a wildland seed collection program, which we'll be talking about today as part of the Willets project. We have a nursery program where we're growing out hundreds of thousands of plants, all source identified, all designed to help support habitat restoration. One of the great things that we have as well is a fully integrated seed cleaning operation where we have the ability to clean seeds at small plot seeds like for wildland collections, as well as large seed productions coming out of our fields. A byproduct of our native grass production fields is native grass straw, which is a great tool for habitat restoration. And we're doing this work with many different partners and collaborators, including many folks from the Department of Interior, including the BLM, the U.S. Fish and Wildlife Service, and the National Park Service, amongst many, many others. And so at the end of the day, what we're trying to do is improve habitat values and restoration success and resilience so that we can improve habitats moving forward. And so at the end of the day, also, our work, we're hoping, will help to solve source identified native seed bottleneck that we're all working so hard to try to solve. So this project, the Willets Bypass Project, is a good example of how we can use individual projects to help us solve that issue. The Willets Bypass Project was conceived decades ago to try and solve traffic issues on Highway 101. Highway 101 used to run through the town of Willets through many different stop signs and stoplights. And a result of that, you'd end up having traffic jams sometimes that were miles and miles long. So for many years, the idea of bypassing the town of Willets came about as a way of alleviating that traffic congestion. But in the process, in order to bypass the town of Willets, it had to go through a very spectacular wet meadow called the Little Lake Valley. And with that, there were significant biological impacts. And in order to approve the project, they had to do significant mitigation requirements, which required lots and lots of source identified native seeds and plants, which is what Heritage Growers is helping to do as part of that project. So we are a subcontractor to the restoration contractor, Hanford ARC, who has a contract with the Mendocino County RCD and who ultimately gets funding through CalTrans for this project. As mentioned, this project has significant biological impacts. More than 70 acres of wetlands were impacted, more than 16 acres of riparian habitat, more than a dozen acres of oak woodland habitat, plus habitat for a couple of special status species, amongst other things. And as a result of that, there were significant mitigation requirements. They had to create more than 50 acres of wetlands. Existing wetland habitat had to be restored and rehabilitated, more than 14 acres of that, more than 250 acres of riparian habitat, almost 50 acres of oak woodland habitat, and lots of mitigation for some rare plant species. And at the end of the day, in order to be able to mitigate these projects, Caltrans had to purchase more than 2,000 acres of land in the Little Lake Valley for conservation purposes, which is the reason why Caltrans used to describe this project as the largest public wetland mitigation project in California. So now I'm going to hand it over to Haleigh. She's going to tell you a little bit about wildland seed collection for this project, and then she'll hand it back to me and I'll talk about the amplification components. So Haleigh.
Great. Thank you, Pat, for that introduction. As he mentioned, I'm the wildland seed collection manager with Heritage Growers, and I'll be talking about our seed collection we did this past summer with this project in Little Lake Valley. So we developed a seed procurement approach with Caltrans that was influenced by what species are available on site, what species have been successful in the past, as well as what additional enhancement is necessary to meet the mitigation goals of this project. And so when it was time to identify the largest and healthiest populations of the targeted species, we worked with a local botanist by the name of Emily Allen, who is able to scout and look for these largest desired stands, which then we would monitor for ripeness to ensure that we found the appropriate window for collection. By the end of the summer, we collected seed that will mostly be directly seeded back in this valley, and then some of that seed will also go to a nursery for propagation. And part of our approach to meeting the required quantities of seed written in our contract with Hanford ARC, we chose to amplify some of the wildland seed that we collected this past summer. And so one of the many cool things about this project that Pat hinted to is that we were given permission and supported by Caltrans to actually collect other seed off of the target species list that are known to be successful in restoration projects, as well as do well in an amplification setting. And then a couple other species that we wanted to experiment with that we also know we want to have in restorations as well, and amplify those so we can support other restoration projects in the region. And so this photo here is, of course, of Little Lake Valley as an aerial view. And then now here we are in ground level, Little Lake Valley. It's a beautiful and spectacular place to roam. It's comprised of oak woodlands and wet meadows. It's right directly in the Pacific Flyway. And so you have a large diversity of birds and raptors that utilize this land for forage and nesting. And then we have the local ranchers who also use this land to herd their cattle, feed their cattle, and manage some of the invasives that still are present on that land today. And surely it's not a true field day if you haven't stumbled across any of the many herds of elk that call Little Lake Valley their home. So back to the seed. We collected seed for 2 different mixes. This is one of the first mixes here. It's called the remedial dry mix. It's mostly comprised of grass and grass-like species, with the addition of 2 species from the aster family, such as the Euphemia and the Symphotrichum. And given that all of these species have a relative similar shape and size to each other, we collected all of these species in a similar method, and we used the grape knife method. If you're not familiar with a grape knife, it's kind of like a handheld sickle. What we would do is we would grab a handful of material, we would cut it at the bottom of the seed heads, throw it in a bucket, and do that as many times as we possibly can in a day or over several days. So this remedial dry mix is going to be seeded in parts of the project site that aren't as inundated as long as other parts of the land. But just because we're calling it a remedial dry mix doesn't necessarily mean that these species are dryland species. And in fact, they're wetland indicator species. It just shows and reflects the large water capacity that this valley holds. The other mix that we collected seed for is this wet mix. As you may have guessed, this mix is going to be seeded on parts of the valley that are inundated for a much longer period of time. And because we have a larger diversity of shape and sizes of plants in here, we used a couple different collection methods to harvest these species. Just like I mentioned in the past slide, the juncus and the hordeum was collected with the grape knife method. But when it comes to the persicaria, the Siam suave, and the pleuropogon, we used a stripping method where we would strip the seed off of the main plant. And that was super efficient, especially when the seed was perfectly ripe and ready, because it would fall just right off of the plant. Another method we used was just cutting the seed heads off of the plant, and we used that method with the cyperus, shown below. And then we have this single species mix, which is kind of a mixture of species we collected that will be individually seeded on site, as well as species that will be going to the nursery for that propagation. And those potted stock that's coming out of those nurseries will be then eventually be planted as high density plantings around the valley. And some examples of those species for those high density plantings is the Euphemia, the Artemisia, and the Symphotrichum. As you may know, all of those species are rhizomatous species, which allows them to be more successful in these plantings. So once you've collected your seed, you go on to your next step of processing that wildland seed. But before we talk a little bit about the importance of doing that appropriately, I want to hone in on this middle row right-hand photo. Right here, we have a collector in a wet meadow and going to town and collecting some seed. Right underneath that tree line, you may see two different invasive grasses growing aggressively. And this wet meadow is comprised of a large diversity of plants. And the point I want to make here is an awareness of avoiding cross-contamination when you're collecting seed. There's no doubt that it's always going to be possible and probable that you will have other species jumping into your harvest bag here and there. But over the years, we've noticed that it's a lot more efficient and a lot easier to avoid that cross-contamination when you're actually collecting in the field, rather than trying to clean it out when you're cleaning that material you harvested. And so taking a little bit of time, a little bit of patience, and a little bit more awareness while you're out there to ensure that your bag ends up being a high quality bag with the material of as much as you can a homogeneous species is important for the process of starting with good quality seed. So once you've collected your material, depending on your initial moisture content, you'll need to dry out that seed for 1 to 6 weeks. You're going to target a moisture content of less than 10%. And depending on the amount of space that you have to actually dry out your material, you're going to want to fluff or turn that material on a consistent basis to allow for good airflow within that and even dehydration. And this proper dehydration is essential for effective cleaning and storage of your seed. And so here's a couple machines that we use to clean our wildland stock seed. The two photos on the left we call a desktop clipper. And this machine separates out the plant material or chaff from the actual seed itself. And then sometimes we take that seed and we put it in this seed aspirator pictured on the right, where the heaviest seed will stay at the bottom and the lightest seed will blow to the top and enter to those two chambers. And any other additional chaff that wasn't pulled out of the desktop clipper will also blow out of that heavy viable seed. And then once we clean our seed, we test our wildland seed to kind of see what we're working with in regards to viability, the purity of it, any weed content that's shown in there. And that directly influences how we choose to seed that wildland stock into our amplification fields. Okay, with that, we're going to play a little bit more musical chairs. I'm going to hand it to Pat.
Okay, thanks, Haleigh. Okay, so yeah, once we have the wildland seed that's been collected and cleaned and tested, then we go with our amplification approach for the project. And so for this particular project, we're amplifying about 15 or 16 different species. The amplification approach includes direct seeding whenever possible. When you have enough seed to be able to direct seed, you always get a better result with that, and it's much more cost effective. But also, sometimes there's not enough seed to go around. And so what we have to do is we have to grow that seed out into nursery plots, into plugs, and then plant those plugs out into the field. We do that both with larger plots, which in this case are 1/10 of an acre, sort of the lower end of the larger plots to several acres for some of the smaller plots. And then we do the same technique here with our demonstration garden where we're doing small plot amplifications and also trying to learn how to amplify some of these species that have never been amplified by us before in the past. And as Haleigh mentioned, one of the great things about this project is the fact that Caltrans has allowed us to be able to go and collect additional species that are not on their list that we've collected and we're now amplifying, things like Elamus glaucus and Dischampsia cespitosa, for example, that now we're growing out, we're amplifying on our own time to be able to create new ecotypes and new species that are available commercially to support habitat restoration in the region. So kind of a nice extra sort of bonus from this particular project. So in the amplification process, it starts out with the field layout. It's really important when you're trying to produce source identified seed is to minimize any cross-contamination of different ecotypes, right? So you don't want to have ecotypes of the same species planted too close together. So we try to lay out the fields in a way that minimize potential cross-contamination. It's kind of a long process and a process that's done over and over again until we get to the point where we've done a reasonable job of having good separation between ecotypes of the same species. Most of the time, we try to seed with our native seed drill. It's a custom-made native seed drill that we had built by Mr. Jim Truax in Minnesota. It's designed to work with our operation, which is on 60-inch beds, which is very different from a wildland seeder. The drill was manufactured. It's able to do 3 beds at a time. So each of these 5-foot wide beds that we have are drilled at the same time. So we have basically a 15-foot wide swath of material that's seeded. Once that drill was built, we had that shipped out to California, out here or to our seed farm, and we had a local machine shop go and build a frame around that, and then add a 3-point hitch for a tractor. And then essentially now we have this precision 6,300-pound drill that does a fabulous job sort of establishing our seeded material within our amplification plots. But as I mentioned, in some cases we have to go out and plant plugs in order to establish our fields because there's not enough seed available to be able to seed. It's a pretty intensive process. The plugs are planted at about 26,000 plants per acre. Essentially doubles the cost of the establishing the field because of all the plugs that have to be done. But it's the way that amplification works with limited stock seed, which is an issue, of course, with when you're trying to do seed amplification of source identified material. So when we're growing out our material, there's 2 primary field maintenance activities. It's irrigation and weed control. So we use sprinklers to germinate the seed that we plant in the fall. And then once that seed is established, we switch over to buried drip tape, which is a great way to establish the seed and save water in the process. But the most important and most intensive part of growing out source identified native seed is weed control, right? So we put in a lot of effort here with weed control. We use cultivation whenever we can. We have to use herbicides in order to be able to control those weeds. We use grass-specific herbicides when we're growing out our forbs, so it kills the grasses, and forb-specific herbicides when we're growing out our grasses. But by far, the most intensive part of the whole process is labor crews. We have labor crews that come out almost continuously removing weeds from our sites. It costs, in some cases, more than $1,000 per acre just in labor crews controlling weeds out in our sites. For the harvest, sort of the most important part of the process, the most technically difficult part of the process is calling ripeness, and that is making a determination of when it is time to take that seed. It's important that you don't harvest the seed too early, because if you harvest it too early, the viability is going to be low. If you harvest it too late, you risk that seed falling on the ground. So it's really important that we sort of harvest this material at the right time. Usually we use two pieces of machinery. The first step in the process is the swather here in the upper left, and in the middle left here, the swather takes the material, cuts it down a few inches off the ground, and creates a windrow, which is a linear pile of field material. What we do then is we carefully monitor the moisture content of that windrow, and just as soon as that's dry enough to thresh, we bring in the combine. The combine then does this remarkable job of separating the seed from the chaff. The idea there is you want to get as much of that material out the back of the combine without losing appreciable amounts of seed. Our assistant farm manager, Mason Schwartz, who also runs our cleaning operation, he's so good at cleaning seed that he sets the settings within the combine so that it works, so the material that's produced and ends up in the tank of that combine can be efficiently cleaned as part of the process. So that the cleaning really starts with the combine. But we can't always use the swather and the combine. We do these small plot amplifications. We have to use small pieces of equipment. Sometimes we have to use things like a sidearm mower on a tractor, or other times we have to do things like use pruning and other things like that. But oftentimes we actually have to go and harvest the material by hand. And milkweed is the classic example of what we have to do with that. So milkweed pods, they differentially ripen. And so because of that, if you want to have if you want to maximize the yields, you basically have to send out those labor crews out to harvest those pods. Sometimes it takes more than a month just to harvest a milkweed field. So it's a very intensive and very expensive process in order to be able to maximize those yields and utilize that hand harvesting. Once the material has been harvested, we put it out onto tarps in that hot, dry California sun to get that moisture content, as Haleigh mentioned, down to less than 10%. Usually it ends up being about 6 to 7%. And by that time, that material is then ready to be brought into our cleaning mill. And here's our cleaning mill. Haleigh talked about the desktop clipper on the left and the small aspirator here in the center. We use these for small lots. But the majority of the time, we run the seed through our large mills, which are actually two mills that are side by side. That seed runs through 3 different machines. The first cleaner uses a series of agitating screens to be able to separate the seed from the chaff. That material then goes through what's called an indent, which separates the seed by shape. And then finally it goes through what's called a gravity table, which separates the seed by weight. We want to hold on to that heavy seed and discard that lighter seed. That heavy seed is going to have the high viability. And then finally, the last step in the process is seed testing. We think it's really important to use high quality seed labs. We want to make sure that the test that comes out is reflective of what's in the bag. Those tests, primarily we're looking at the purity, the viability, the number of live seeds, and then finally the PLS, or pure live seed, that comes out of that test. And that information informs things like seeding approaches that we'll use out in our fields for amplification. And of course, it'll help CALTRANS in use in putting together these wet and dry mixes that Haleigh mentioned before, as part of their process to make sure that they're using the balanced seed approach that maximizes the use of that seed. And so that's it. Just wanted to mention here that hopefully that QR code, you can see that on the right-hand side. You can click on that and it goes to a blog that talks about the restoration work that's being done at the Little Lake Valley. Gives you some additional information on that. And I think with that, I think we probably have time for some questions.
We do, yes. Thank you, Pat and Haleigh, for that presentation. We've got quite a few questions in the Whova Q&A box. So if you'd like it answered, please upvote it and it can be moved to the top. So the first question that we have for you is, are collections happening on public, private, state, or federal land? And if it's happening on state or federal land, what does the process of getting permission look like for you to be able to do harvest and amplify from that harvest?
Yeah, so in this particular case, the harvest is on state lands. Caltrans owns these lands. They had to buy these lands in fee title as part of the restoration. Because in this particular process, we're doing the work for Caltrans. Essentially, once we were awarded the contract, essentially just working with a local biologist, Marine Doyle, to allow us to be able to collect that material. Now, depending upon other seed collections, sometimes, to be able to get permission to collect on private lands, it could be as easy as an email. But on other lands, like the BLM, for example, if you want to collect on their lands, you have to go through a permitting process. So it kind of varies. It depends. So, but no real formal process here as part of the Willets project. So.
Great, thank you. Okay, next question. Willets has a long history of seed collection and restoration. Many of the sites have been seeded with natives in the past. How do you avoid collecting seed from areas that were recently restored, like not capturing truly wild genetics?
Yeah, that's great. Excellent question. In fact, Haleigh mentioned the other day how actually some of the restoration sites are doing so well that she has to be very careful in where she's collecting the seed to make sure she's not collecting within these restored sites as part of it. But Haleigh, there's lots of maps that are part of that are available. And one of the things about this is that in the event that she did actually accidentally sort of venture into a part of a restored site, all the seed that was used for restoration over the years at Willets has all come from the site. So it is all local genetics, if you will. So.
Great, thank you. Okay, next one up for you is, what was the former land use of the 2,000 acres that Caltrans took over as mitigation for the project?
I think primarily grazing.
It's primarily grazing is what I've seen.
Yeah, and part of the conservation efforts out there is managed grazing, right? So managing in a way that sort of helps to maximize conservation values. Like a lot of that meadow foam mitigation actually is doing conservation grazing. The meadow foam mitigation is doing conservation grazing to try to establish habitat conditions that work with that species. Yeah.
Great, thank you. I think we can squeeze in one more question. Do you use fumigation or other methods to treat wild seed for possible insect infestation to prevent seed destruction during seed storage?
Yeah, so far we haven't found insects in our seed, but if we do, we will have to use fumigation. So that's just part of it. So that is something we'll probably end up having to do in the future, but so far we haven't.
Great, thank you. All unanswered questions will be moved to the HUBA Q&A box, so don't worry if your question wasn't answered. Thank you again, Pat and Haleigh, for that great presentation, and I'll pass it to Alexis to introduce the next speaker.
Thank you.
Yeah, great. Thanks, Pat, and thanks, Haleigh.